Hello, class. I hope you're having a great week. First, I would like to thank all of you for all the feedback you received, and as one of you mentioned, that I didn't realize that you are having a data issue. So therefore, the request was to upload videos to YouTube. That way, because I mean, most of us have unlimited packages, so that way, there will be no data cost or data penalty. So therefore, I already uploaded lecture number 3, right? Oh, two. Yeah, 2. Yeah, to the YouTube, and I hope you saw that. I also sent you an email indicating that I already uploaded the video to the YouTube. So therefore, it's always very good that you guys communicate. So therefore, we can keep on improvising to provide you the at least the near perfect situation for your studies. And I'm hoping that you guys are planning to come back to the university within a couple of weeks, since as I heard, the strike might be over because of the election, but you never know. So we'll see. Anyway, so last week we discussed about the light reaction and also a general overview about the photosynthesis. And I hope all of you had a chance to go through all the additional reading I gave you, and to learn more about this beautiful process of photosynthesis as well. So moving forward, so we are going to continue talking about the photosynthesis, and not forgetting our friend as well. So we'll see what we are going to talk today. And I hope all of you had enough time to go through all the content I uploaded and all the recommended reading. And I think probably now you're appreciating even more about this, the process of photosynthesis, right? So moving forward, I'm going to talk one concept before we moving into the second stage, because I think it is very important for us to learn about this very, very unique and important characteristic. So this is what we called as quantum yield. I think you might heard this term before, probably during your A levels, but anyway, I'm going to revisit and then try to introduce you this concept of quantum yield. If someone asks you what is quantum yield, so basically we are going to tell that specifically when we talk about photochemistry, so the quantum yield of photochemistry is basically a number of photochemical products produced as a result of the energy or the quanta that was absorbed by our photosynthetic apparatus. In other way, it's basically you divide the number of photochemical products by the total number of quanta absorbed. It will give you the value, or basically the ratio that can be interpreted in order to tell people about what is the yield actually we obtained or we received as a result of our photochemistry. But before we jump into the other aspect, so I wanted to mention one thing. So that is, if you think about the oxygen production, remember that the water was splitted and then oxygen was released. If you think about the quantum yield of oxygen production in our photosynthesis, it is about 0.1. Sometimes it's range from 0.1 to 0.3. Okay, so seems like only 1/10 of the quanta absorbed is used for the oxygen production. In contrast, if we looked at our photochemistry, because photochemistry is the most important thing for us, right? If you think about a functional chloroplast under dim light, that means without saturating condition, so the photochemistry of it's going to, the quantum yield of photochemistry is going to be around 0.95. So that is a very good yield, right? And if you think about the fluorescence, remember we talked about fluorescence, because when the energy is received by chloroplast or the chlorophyll molecules, so they have 4 things they can do. So we can see photochemistry is around, the quantum yield of photochemistry is around 0.95. And if you think about the fluorescence, the quantum yield of fluorescence is 0.05 or lower. And if you think about the other processes, they're actually negligible. We can't even get a good value to calculate the quantum yield. So that tells us tells us that photochemistry is the most common result of chlorophyll excitation. So that is good, right? Because that means there are 4 processes. They're competing for this, the energy change, and among them, the photochemistry, the most important for us, is going to have the highest efficiency. That represent, that basically turning into a value of 0.95, the quantum yield of 0.95. So that means this is one of the very efficient reactions you see in the nature. I hope it is clear. So now we can move on to another interesting phenomenon called a red drop effect. Let's see what this red drop effect is. Okay, and when scientists looked at the spectrum, remember we talked about the visible spectrum, and I mentioned that visible spectrum is used for the photosynthesis, usually ranging from 350 to 700, or sometimes we call from 400 to 700, which is what we call as photosynthetically active radiation, or PAR. And there, so remember you learned about the absorption spectrum and action spectrum. If you look at the absorption spectrum, you can see it goes like this. Okay, so you can see there's 2 peaks, and then around blue region you have high absorption, and also near the red region you have a higher peak of absorption. And if you look at the quantum yield of the photosynthesis, what you're going to see is, so basically from 400 to 650, it fluctuates but not much. But the moment it goes beyond 680 over bar, you can see there's a dramatic drop. And this is let me get a pen and let me see there, you see there is a drop, and scientists were puzzled by this phenomenon, and they name it as red drop effect. Okay, and let's see how we can basically dissect and try to understand what is happening what is actually happening in this reaction. And remember I told you about immersion last week. So this was discovered with the help of the immersion experiment, or what we call as, and we name it as in memory of him, immersion enhancement effect, or in simple terms, enhancement effect. What he did is very, very intelligent thing. He measured the rate of photosynthesis separately. What he did is he used 2 wavelengths of light, okay, separately. And then instead of using separate wavelength, he used both at once. Okay, so he was doing this experiment for different wavelengths. What he found out was, when he used red and far red light together, he observed the rate of photosynthesis is going to be more than the sum of individual beam lengths. So that means basically our photosynthesis, the photosystems, they absorbing red and far red. And now this explains why we observe the red drop effect, because the moment you go beyond, what is going to happen? There is no photosynthesis happening. So therefore, those wavelengths, so they carry less energy, because the higher the wavelength, lesser the energy. So therefore, you see dramatic drop in our quantum yield. So this is what we call as red drop effect. Okay, so if we discuss more in detail, the photosystem 1 preferably absorb far-red light of wavelengths greater than 680 nanometers. And photosystem 2, it preferentially absorb red light of 680 nanometers. And this photosystem 2 is driven very poorly by the far-red light wavelengths. Okay, and as I mentioned to you earlier, this wavelength dependence, we call it as the enhancement effect, or the red drop effect, or immersion enhancement effect. Okay, so our photosystem 1, as you can remember, it produces a very strong reductant, and this strong reductant has the ability to reduce NADP plus and make a weak oxidant. And if you think about the photosystem 2, it's quite the opposite. What it does is it basically produces a very strong oxidant capable of oxidizing water, and weaker reductant than the one produced by photosystem 1. Okay, so therefore this is something you need to remember, because it is very important. Okay, so with that, we are going to move into the carbon reaction. Remember, last week I mentioned to you that we are not going to call it dark reaction. Rather, we will call it as carbon reaction, because I will explain to you in a bit. There are some light dependent component when it comes to the so-called dark reaction. So therefore, you can't call it as a dark reaction. Okay, so let's see how our light reaction is going to link with the carbon reaction. Take a closer look at this diagram. As you can see, so we have our light driven reaction. So there, if you can remember, they produce NADPH and ADP. What is going to happen is, so this NADPH and ATP will basically provide input for our carbon reaction, which is happening in the stroma, and in result it is going to produce carbohydrates using carbon dioxide and water. Okay, in very simple terms. And I think during your A levels you learned that this cycle as Calvin cycle, but it was later named as Calvin and Benson cycle, because Calvin was it was discovered in Calvin's lab, but due credit was not given to Benson, one of the other researchers involved in this project. So nowadays we call it as Calvin, Benson and Basham cycle, because they were third graduate student. So once you graduate from this degree, so some of you will go and do your postgraduate degrees. So those are what we call as graduate students. And then, so there were three people behind the discovery of this cycle. And now that's why we call it as Calvin, Benson and Basham cycle. Okay, so now let's dive in and take a closer look at the carbon reaction. In very simple terms, we can divide this carbon reaction into three separate, how can we say, three major steps. Okay, so the first step, what is going to happen is what we call as carboxylation. And then the second phase, the reduction is going to happen. And in third phase, the regeneration is going to happen. Why we have a cycle instead of a linear reaction? Because then imagine, so if you have a linear reaction, every component needs to be produced, right? There's no regeneration. So therefore, once we go this way, so then you have to make things from here to here to here. But if you have a cyclic reaction, what we can do, so and it can continuously happen without many interruption. So that's why most of the biological reactions, the essential ones, so they are cyclic in nature. So that way, so at the end of the reaction, so part of the output is going to basically act as input for the beginning of the reaction. So therefore, that's why it's a cyclic process. And as you can remember, so the starting point is ribulose 1,5-bisphosphate. What is going to happen is it's going to react with carbon dioxide and water. So that's where the carbon dioxide is going to interfere interfere with the reaction, and it is going to produce 3-phosphoglycerate, and then 3-phosphoglycerate will be converted into triose phosphate and dihydroxyacetone phosphate by using ATP and NADPH, and then some of the outputs of this reaction will go into our carbon output, and the rest will be used in order to produce ribulose 1,5-bisphosphate again, and it is energy consuming reaction. So this is what we call as Calvin, Benson and Basham cycle, and what is happening inside. So we will have a quick look at individual components or an individual process, carboxylation, reduction and regeneration in next slides. So let's look at carboxylation, and as you can remember, the starting point is ribulose 1,5-bisphosphate, and it's going to create 3-phosphoglycerate through the process of carboxylation, and CO2 is going to provide the carbon input. And here the CO2 acceptor molecule is going to be the ribulose 1,5-bisphosphate, and when it reacts with carbon dioxide and water, it's going to produce 2,3-phosphoglycerate, because here you have 6 carbons, and then the outcome is a 3-carbon compound. So therefore, you're going to produce 2 3-phosphoglycerate molecules. Okay, so in simple terms, a 5-carbon acceptor molecule is going to create 2 molecules of 3-carbon intermediate during this process, and this process is driven by our carbon input. Okay, so the next step is the reduction. What is going to happen is this 3-carbon intermediate molecules or 3-phosphoglycerate, and these ones will be converted into triose phosphates, or in simple terms, 3-carbon carbohydrates, as you can see here. And this reaction is driven by photochemically generated ATP and NADPH. Okay, so that the input from our light reaction is going to provide the driving force for this reduction process. And then whatever a set of output is going to go for the production of starch and sucrose, and that provides the carbon input for all living organisms. And then one of the crucial step is what we call as regeneration, and there we are going to basically produce our CO2 acceptor molecule, ribulose 1,5-bisphosphate, and this basically in simple terms, this regeneration is actually the regeneration of ribulose 1,5-bisphosphate in order to drive this reaction continuously. And you can see it's for easiness, this is like there's only like one step, but in reality, so there are 10 enzymatic reactions involved in this process. So that means there are 10 enzymes catalyzing this reaction, and out of this 10 reaction, one reaction requires ATP. Okay, and to show you like in detail, so this is how it's going to happen. So now I want all of you to remember like all the structures as well as all the steps, okay, and all the enzymes involved in this process. So you guys going to love it, right? Because you guys are really good in remembering things. That's what I heard. So therefore, like I guess you guys are ready, right? Okay, so I'm so proud of you guys, because you guys can basically like remember all of these things. Okay, so I was kidding. No, I am not going to expect you to remember all the structures and like each and every step in this process, because you can always go and like find it from somewhere if someone needs that information. But rather, I would like you guys to understand the process and like and the main steps, like as I summarize, and most importantly, so I want you to think how you can harness this knowledge about this carbon reaction and how you improve our crops in order to have a much better yielding crops. So that is what I require, not memorizing things like from like all the structures and other things. So therefore, don't worry too much because you don't need to like memorize all these different reactions. Okay, so in summary, what happened was if you put like all reactions into like summarized form, so three carbon dioxide molecules and 3 ribulose 1,5 bisphosphate and 3 water, 6 NADPH, and ATP and everything is going to basically get together, create 6 triose phosphate and 6 NADP plus, 6 ADP and 6 phosphate groups. And then so this intermediate molecules is going to convert into 5 triose phosphates and 3 ATP and water, and then it will be converted into 3 ribulose 1,5 bisphosphate, 3 ADP and 2 phosphate groups, and then the rest is going to convert it into triose phosphate. Okay, and for us, the real deal is basically the production of glyceraldehyde 3-phosphate, because this is what is going into your favorite kottu, because that carbohydrates are important for like food or like food production. Okay, so this is how the carbon reaction is going to happen. Basically, this is like a very quick overview about this reaction. Okay, and if you think about this reaction, so every reaction like is not going to happen like like that, and it's not going to be continuously happening all the time, because if you can remember, photosynthesis is not going to happen all the time, right? Because at night the plant, they are not going to do photosynthesis. So then what is the point activating all the steps involved in these different reactions? So therefore, so cell is not going to waste energy like us. So they always think like 10 times how we can increase our energy efficiency. Okay, so therefore, if you think about the carbon reaction, so this reaction, there is an induction period. In the dark condition, our photosynthetic enzyme, so they are going to have a low activity. So there's like a very small amount, not zero, because otherwise you can't start, no. So therefore, like there's a very low level activity of photosynthetic synthetic enzymes, And also there's like very low concentration of intermediates of our Kelvin, Benson and Basham cycle. What is going to happen when there's light? So when the illumination happens, the rate of carbon dioxide fixation is going to increase gradually. Okay, not like it's going to be a gradual increase. So that is how it is going to happen. Okay, and if you think about how this Kelvin, Benson and Basham cycle, because now we have like a bunch of reactions happening, and we have to have some control, right? And if you think about the compounds, or like involved in the regulation of compounds and forces involved in Kelvin and Benson and Basham cycle, so that includes Rubisco activase, and the light is going to play a major role. One is through the thioredoxin system, and also light dependent ion movements, and also light controls the assembly of chloroplast enzyme into supermolecular complexes where all these different reactions can happen. Okay, and these things basically controlling 5 enzymes, including Rubisco, fructose 1,6-bisphosphatase. And when I said fructose, it's like a small funny story. Fructose is basically like something to do with fruits now, because fruits contain a lot of fructose. So therefore, like if you, here in like British colonies, we call it as fructose. But if you go to North America, so they say fructose. Okay, so it's up to you to pronounce this term. If you go to America, they will laugh at you if you say fructose. They say fructose. Okay, so therefore, like I sometimes use like a mix of it, because when I was studying there, so I had to use the fructose, like vitamins and vitamin, car park and parking lot. So they are like different variations. So it's interesting. Sometimes you will see like they will ask, oh, what are you talking about? And then you realize, oh, you need to change it. Okay, so that's next time when you so probably like most of you will go abroad to do your higher studies and maybe for jobs, and then that is something you guys going to experience. Anyway, so moving forward, the other 3 enzymes are the pseudoheptulose 1,7-bisphosphatase. Then you have phosphoribullokinase, NADP glyceraldehyde 3-phosphate dehydrogenase. So these ones are basically controlled by these 4 things. So therefore, remember, light is basically controlling the so-called dark reaction. So therefore, don't write dark reaction when you answer your exam papers. Last time I noticed that people use dark reaction. Actually, it is carbon reaction. Okay, so I'll probably like put like -2 marks every time you write dark reactions. Okay, so moving forward, so there's like a very interesting thing we are going to discuss here, as well as in like towards the end of the lecture. You will see the enzyme Rubisco. Okay, so Rubisco is the most if you think about like all the proteins in the world, Rubisco is the king. Okay, because it can all the plants contain Rubisco, and it's a big enzyme. So therefore, like it is the most abundant enzyme in the world. And this one was evolved way back. Remember when we talk about like oxygen levels and other things? Remember the early atmosphere, there was less oxygen. Okay, so therefore, the Rubisco, when it was like basically evolved, there's nothing to worry about like oxygen. So therefore it says, okay, I don't care about oxygen. And what happened was, because of the Rubisco, the oxygen level went up. And what happened was, so this enzyme can basically act as, like, it can do both carboxylation as well as the oxygenation. So therefore it creates a chaotic situation, because when there's oxygen, very interesting reaction is going to happen, and that reaction is not efficient as the regular the Calvin, Benson and Basham cycle step. Okay, now, like, until last year, because I was teaching, like, the same course for like 7 times during like last 7 times during last 6 semesters, because there was no synchronization. Like, you like every year there's like in every semester there was like 2,200 batch, and then like food science students, you guys know, you do it in 1-2. So one time we had to offer the same course twice, like in 2 weeks apart, one for ATM student and one for food science student. And like we were like, okay, teaching is fine, but marking papers, gosh, it was like basically killing me and Professor Chandrika, because you guys are very good when it comes to writing. You write everything, and then marking papers make our heads spin, so that's something like that. Anyway, coming back to the Rubisco, until like last year or like last semester, so we were like basically talking about this Rubisco being like a very old school enzyme. But now we have evidence to say that this Rubisco enzyme is slowly getting better. It is going more, or like it's going for higher efficiency towards a carboxylation. Okay, so I will upload a link, so you will see the link up here, and so please check the this news article, and it's also on your eHub as well. So they are like they're talking about how this old school enzyme is evolving, because remember, when you were facing the COVID-19 pandemic, that virus was evolving relatively very fast. You had like different variants, right? You had like omega, you have beta, so you have gamma, blah blah blah, and Omicron, and like I can't even remember names. So therefore, like there was a lot of changes, but this one is evolving like in snail phase. Okay, very very slow, but it is happening. So therefore we have a good hope for the future, but it doesn't mean that you can release more fossil fuel and create more global warming. No, don't do that. But remember that the nature is especially the process of evolution is so awesome that it changed things to match with whatever changes happening in our environment. So what is the bad thing with this oxygenation? I think this is something you learn, and I'm going to remind you about this process. So what we call as photorespiration. Okay, and the photorespiration, unlike the regular carbon reaction, so this one is not going to restrict to the chloroplast. Instead, it is going to basically involve in 3 different organelles. So that includes chloroplast and peroxisome and mitochondria. So that means instead of happening things in just one place, so now it's happening in 3 different places. So that means the efficiency is going to get affected. And if you look at these different processes, the different organelles and reactions involved in this process, you can see. So you have our ribulose on 5 bisphosphatase, and what is going to happen, instead of accepting a carbon dioxide molecule, now it can accept oxygen molecule, and this oxygen molecule will be converted into glycolate inside the chloroplast, and then basically it is going to move in to the peroxisome, as you can see here, and then it will be converted into glycoxylate, and then it will be converted into glycine. Okay, and then this glycine is going to go for a trip into the mitochondria, as you can see here, and then inside the mitochondria it is going to get converted into serine, and then it is going to go back into the peroxisome. Okay, and then from peroxisome the serine is going to convert it into glycerate, and then this glycerate is basically going into the chloroplast, and then through a set of reaction it is going to enter into the Calvin, Benson and Basham cycle. Okay, so this is what is going to happen. So now in Sinhala what we call as Parangya Kote gaya wage. So that means basically like you want to go from one place to here, instead of taking this route, so rather you are going to take like route like this. So therefore the outcomes are going to be different, and during this process it is going to release one molecule of carbon dioxide, and it is going to consume two ATPs and then two molecules of reducing equivalent, and it is going to incorporate 3 carbon skeletons, as I showed you earlier, back into the Calvin, Benson and Basham cycle. Okay, and it is going to restore the glutamate from ammonium ion and from 3-oxoglutarate. Okay, and if you look at like the net reaction that is happening, as you can see here, so we can basically sum up everything into this reaction, as you can see here. So the ribulose 1,5-bisphosphate is going to basically react with oxygen, and also water and glutamate will be converted into glycerate, and then 2,3-phosphoglycerate, ammonium ion, carbon dioxide, 2-phosphate group and 2-oxaloglutarate molecules. And then this 2-oxaloglutarate molecule is going to convert it into glutamate, and then glycerate is going to convert it into 3-phosphoglycerate. Okay, so therefore this reaction is not going to be efficient as the carbon reaction. Okay, so if you look at this one, you can see the chloroplast. So this is what is going to happen. Okay, so you have a Calvin, Benson and Basham cycle there, and then we have our light reaction, and then what is going to happen is like it's going to provide all the energy requirement for the Calvin, Benson and Basham cycle, and then when there's oxygen, the photorespiration is going to happen. Another name for photorespiration is C2 oxidative cycle, as indicated here, and then so eventually everything will come into the Calvin, Benson and Basham cycle. Okay, and the important thing is, although we tend to think that photorespiration is a bad thing, of course it is less efficient, but it's also an adaptation, because so there are some good things related to the C2 oxidative cycle or the photorespiration. Like in one example is basically it is important for the nitrogen cycle, and also there are many other plus or the pros regarding the photorespiration. And there is one document uploaded into your eHub, and also link is given here. What you can do is basically go and read that article and grab the essence of the article, because there might be a question coming into exam. So that's how I motivate you to read. Okay, so therefore and read the article and then see so many interesting facts about photorespiration. That is important. Okay, so I feel maybe you guys are feeling sleepy. Hope not. Okay, so So I'm going to talk more about this the problem, and then how plants naturally came up with different mechanism to minimize or avoid this issue. Okay, so let's see. I will meet you in the second video very soon. Okay.